The focus of this video will be on leukocytes, or white blood cells. After viewing this video, you should be able to identify and understand the following goals. Differentiate good views versus poor views, locate the feathered edge, describe how to scan a blood smear, identify different types of white blood cells, and identify other things in a blood smear. This slide is a review of the video you watched. The feathered edge is at the tip of the blood smear, where the blood appears thinner. When viewing your slide, it is important to move your slide in the pattern on the right image to prevent double counting or missing elements in the smear. Here are examples of viewing areas. The example on the left is good as there is space between the cells and they are not overlapping, while the right view is the opposite. It is important to have a good viewing area to be able to distinguish between red blood cell morphologies, blood elements, and to detect anything abnormal in the smear. The image on the right is a poor view, where you are drifting away from the feathered edge and the cells are too squished together in the viewing area. Now we are going to discuss the different types of leukocytes. In a lab, a white blood cell count, or differential, is done. This is a process of identifying the different types of leukocytes and counting how many there are of each, usually in 100 total leukocytes. There are two categories of leukocytes. The first are mononuclear agranulocytes, or cells with a single sphered nucleus and no granules in the cytoplasm. Lymphocytes and monocytes are in this category. The second category are polymorphonuclear granulocytes, which have lobed nuclei and granules in the cytoplasm. Examples of these include neutrophils, eosinophils, and basophils. Neutrophils are more frequently found, usually about 50 to 65 per 100 leukocytes. Neutrophils have a purplish-red nucleus and a right-stained smear that will have 2 to 5 lobes called a segmented nucleus, or seg. However, the nucleus of younger neutrophils may instead be banded, looking like a C. The granules in the cytoplasm are small and look like red or purple dots. The purpose of neutrophils is phagocytosis, or eating bacteria. Neutrophils also secrete chemicals to enhance an inflammatory response. Lymphocytes are the second most common leukocyte, with about 25 to 40 being found in every 100 leukocytes. A normal lymphocyte has a single lobed nucleus that is dark purple in a right-stained smear, and it is usually round, appears solid, and is generally smaller than other leukocytes. Little cytoplasm is left behind because the nucleus fills so much of it up. On the right is a reactive lymph, which appears to be stretched out similar to silly putty. Reactive lymphs are normal to find. Lymphocytes are important because they secrete toxic chemicals, and some are more directly involved with an immune response. Monocytes are the largest leukocyte and appear blue in a right-stained smear and have a single lobe nucleus that may sometimes appear folded over itself. The cytoplasm is a dusty gray-blue color and may contain vacuoles, which appear as small white circles within the cell. Generally, you will find 0 to 4 monocytes per 100 leukocytes. The function of monocytes is phagocytosis, but since they are large, they are able to phagocytose larger bacteria, and they also pick up cellular and tissue debris. Vacuoles are additional sites of digestion within a monocyte. Eosinophils have a double-lobed nucleus that appears purple in a right-stained smear. The cytoplasmic granules are large and orange in color. You will generally find 1 to 3 eosinophils per 100 leukocytes. Eosinophils secrete toxic materials to kill invaders, phagocytose bacteria, and also secrete chemicals in allergic reactions. Basophils are found the least frequent in the blood, and none to 1 found out of 100 leukocytes is normal. In a right-stained smear, basophils have a light purple C-shaped nucleus. However, it is often not visible because of the large, dark purple granules in the cytoplasm. Oftentimes, you may see ghost granules, which appear washed out and barely visible because they do not stain well. Basophils release histamine and heparin, both of which are important in inflammatory responses such as allergies. Additionally, in a blood smear you will see red blood cells, which look like red or pink discs, and platelets that appear as small blue specks, which you can see in the picture on the right. It is important to recognize what these look like because you may have to perform red blood cell or platelet counts. To wrap up this video, please remember to know good views versus poor views, know how to locate the feathered edge, know how to scan a blood smear, differentiate the types of white blood cells, and also identify the other things in a blood smear. If you have any other questions, feel free to bring them up in lab.